Hello everybody. In today's lecture we are going to discuss comparative inference procedures for single channel microarray data when more than two types of mRNA samples are under study. So again I will discuss one case study and explain all the concepts and the procedures via the case study. So in this case study we have three groups of obesity. One group is non-obese, another one is obese, and third one is medium obese. What's the aim? Aim is to identify the gene which are responsible for obesity as well as for non-obesity. Okay, because that is of most importance. So in the study, 8 individuals from each of these three groups were identified. 8 non-obese individuals, 8 obese individuals, and 8 medium obese individuals. And then mRNA was extracted from the cells of these 8 individuals from each group, and it was hybridized separately on 24 arrays. So there are 24 samples, 8 from each category, and there are 24 arrays. So on each array only one type of sample was hybridized. So this is a single channel microarray data, and we'll see how this type of data is analyzed. So the data available was expression values of 22,283 genes and on 24 arrays. So that's the data matrix we have to deal with, and our aim is to use a kit of statistical tools to identify a handful of genes out of these 22,000 genes which have a distinct profile in non-obesity, obesity, and medium obesity. Okay, as in the previous lectures we have discussed that the first step is always go for a transformation, because the histogram and box plot of expression values on any one of these arrays gives us this picture where there is a huge bar at the lower intensities and almost negligible bars at the higher intensities. The box plot is, one has to literally tell us that it's a box plot because of the many genes having expressed at the low levels. This is bound to happen because of the experimental setup, because all these expression values range from 0 to 2^16. So there is heavy clump at low intensities, and it's very difficult to interpret this data and analyze such a data when there is a huge variability of this order. So the first step is always go for a log transformation, and again because the original range of the data is between 0 to 2^16, we go for a log transformation and we select base as 2 and see the histogram and box plot of the transformed data. Now the box plot is visible. Again, what we interpret from the box plot is the line, the middle line which represents the median is at the below the lower half. So again it's a positively skewed data, but that is immaterial. We will not go into the details of this. So the first step is, as I told you, the log transformation to the base 2, and then the next step we always go for a box plots of all the expression values on all the 24 arrays. Here is a picture of box plots of 8 arrays which correspond to the non-obesity. So that NO1, NO2 and so on up to NO8 indicate that these are the gene expression values from 8 arrays coming from the non-obesity group. What we notice here is, now remember it's a single channel microarray data. So only one type of mRNA sample is hybridized on one array, and there is no question of dye bias. So whenever we are in a single channel microarray data, we need not worry about the dye bias. It's not present at all. What we have to worry about is the variation due to the artifacts which enter in due to the various stages of the microarray experiment. Again, when we look at this box plots, what we notice is more or less the box plots are similar. Their medians are also similar. The variation, the width of the box plot tells us about the variation. It is more or less similar. One can always quantify this similarity by what are known as concordance coefficients. So concordance coefficient, if they are less than 0.8, it is necessary to go for a normalization. If they are enough high, above 0.9, then one may not go for a across array normalization as well. So the picture for these 8 arrays is distribution. Its empirical distribution of expression values is slightly different. One can also find out concordance coefficient, which gives us the numerical measure of concordance. And then for this data, it was found that concordance coefficients across arrays are quite high, larger than 0.9. So one can even skip quantile normalization. Even if we go for quantile normalization, no problem. It doesn't take much time even to implement. So without going into this concordance coefficient, one can always proceed with the quantile normalization and work with the normalized data. But it can be skipped in some cases. This may not be the picture for all the experiments. Alright, so for this obesity data, one can skip the quantile normalization, and then the microarray data that we had in our hand was of the type. We had a gene expression levels as the body of the matrix. First column gives us the gene annotation. These are gene identities of 22,283 genes. First row gives us the information about the samples. So first 8 columns are corresponding to non-obese group. Columns number 9 to 16 are corresponding to obese group, and next last 8 columns are corresponding to medium obese group. How many rows we have? 22,283 rows corresponding to the genes. And aim is of course to identify the genes out of these thousands of genes which have different conditions, different profile under these 3 setup. Okay. So again what we do is we'll try to implement appropriate inference procedures to identify the genes which have different profile. So going back, see what is that we have to now study. These 24 columns apart from this first column, okay, that is the gene identities. So main data matrix has the expression values in 24 columns, and then what you have to do is whether the gene expression values in these 3 groups of columns, each consisting of 8 columns, are similar or not. So going to the statistical language, what we want to test is whether the entire genome of non-obese group, obese group and medium obese group, it's the same or whether it is different. So null setup always is they are same, but then comparing genome is comparing the gene expression values of all these 22,283 genes. And therefore the first step is we divide this entire family of hypotheses into the hypothesis corresponding to each of the gene. So for each of the gene we have to test the hypothesis of the type mu1 equal to mu2 equal to mu3. What is mu1? Mu1 is the gene expression value, average gene expression value in population of non-obese individuals. Mu2 is the average gene expression value in the population of obese individuals, and mu3 is the average gene expression value of median obese group of individuals in the population. So that is what we want to test. So our null hypothesis is mu1 equal to mu2 equal to mu3 for each of the gene. Alternative we always select as the two-sided hypothesis, that at least one of these mu1, mu2, mu3 are different. because we don't have any prior knowledge about the direction of these expression values on the population level corresponding to these three groups. So alternative is at least one of these three groups differ. In such a setup, the immediate tool that comes to our mind is the one-way analysis of variance, but we can always go to this tool provided the underlying assumptions are satisfied. So we always begin with verifying the underlying assumptions and then select the appropriate test procedures for testing the hypothesis mu1 equal to mu2 equal to mu3 against the alternative that at least one of these mu i's differ. Okay, so the first step in our analysis is we test normality for each gene using 24 observations. Now let me explain you one more thing here. These 24 observations corresponding to one gene, that is the first row of our data matrix, consists of three groups of observations. First 8 corresponding to one group, second 8 corresponding to the second group, and third 8 corresponding to the third group. So actually they are not on equal platform, their means can be different. So what we do is usually we scale the data, scale in the sense that we make their mean 0 and mean is 1. We standardize it and then apply, then bring all these on equal level and then apply test of normality on all the 24 observations together. If at all the normality is rejected, it must have been rejected in at least one of the groups. So we cannot use our one-way ANOVA analysis for that. If it is accepted, then it follows that all these 24 observations are coming from normal, and then it tells us from our theory of multivariate normal distribution analysis that each of these 8 must have been coming from a normal distribution. So using this setup, using the scaling procedure, we can group all these 8 observations coming from 3 groups to be together and increase the number of observations for testing the normality. Alright, so we apply the Shapiro-Wilk test on each of these 22,283 genes using 24 observations on each gene separately. The result was normality was accepted for 16,400 genes and it was rejected for remaining 5,883 genes. So when it is rejected in at least one of the groups. So normality is rejected at least either for obese group or non-obese group, or maybe more than 2 groups. So we cannot go for our usual parametric test procedures. If it is accepted for all these on the basis of 24 observations, it is accepted for each of the groups separately. Alright. So for those genes when the normality is rejected, we apply Kruskal-Wallis test. It's a non-parametric version of the one-way ANOVA, an extension of Wilcoxon rank sum test and so on. So we apply Kruskal-Wallis test on all these 5,883 genes. The result is, out of these around 6,000 genes, the hypothesis of equality of means is rejected for 40 genes at 1% and 321 genes at 5%. So these 40 genes are definitely differentially expressed in the 3 groups of obesity, and these are the genes of our interest. Well, now let's go to the set of genes for which normality is accepted. The second assumption that we have to verify if we want to apply one-way ANOVA test procedure is whether the population variances are same for all the 3 groups. So on all these 16,400 genes, we apply the test for equality of variances, and that's the well-known Bartlett's test. So it is applied on all these, and the result is equality of variances is accepted for 15,636 genes, and it's rejected for the remaining one. So when the normality is accepted and equality of variances is also accepted. Both the assumptions underlying one-way ANOVA test procedures are satisfied. So on this group of genes, on 15,636 genes, we apply one-way ANOVA test procedures. The result is for 177 genes the equality of means is rejected at 1% level of significance, and for 840 genes it is rejected at 5% level of significance. Thus again these 177 genes are of our importance. These are the genes which have a different profile under obesity, non-obesity and medium obesity groups. So these are the genes of our interest which needs to be further investigated. A second group of genes for which normality is accepted but the equality of variances is rejected, we apply a modification of one-way ANOVA test which is known as one-way ANOVA Welch-Ebb test. It's again the extension of two-sample Welch-T test. What are the results? Out of 764 genes on which we applied this one-way ANOVA Welch-Ebb test, for 19 genes the equality of means is rejected at 1%, for 67 genes it's rejected at 5% genes. So these genes are again of interest because for these genes the profiles under three conditions is different. Alright, so what we get from these three test procedures, Kruskal-Wallis test, one-way ANOVA test and one-way ANOVA Welch-Ebb test, we are able to get a set of genes which have a distinct profile in three conditions. But we have to go further here. We want to know that it's what we get is that mu1 equal to mu2 equal to mu3 is rejected. So maybe mu1 is same as mu2 or mu1 is same as mu3. So the profile is same in obese and medium obese group and it's different in non-obese group. So to identify this, now we have to apply pairwise test procedures for on all these genes for which the equality of means is rejected. So we proceed with appropriate pairwise test. So on these 40 genes or 321 genes which are rejected according to Kruskal-Wallis test, we apply pairwise Wilcoxon-Rank-Sum test to find out which genes, whether the gene has a different profile in which group. So there are three pairs of hypotheses that we have to test: mu1 equal to mu2, alternative mu1 not equal to mu2, second is mu1 equal to mu3, alternative is mu1 not equal to mu3, mu2 equal to mu3 and alternative is mu2 not equal to mu3. So these three pairs we need to test, and then the result is shown in the next slide. Similarly, for one-way ANOVA we apply pairwise t-test for these three contrasts that we have to test. For one-way ANOVA Welch-Ebb test procedures, we apply corresponding pairwise Welch test. So these pairwise tests again tell us that the gene has a different profile in which group. Okay. Along with the results of these p-values of pairwise Wilcoxon-Rank-Sum test, pairwise t-test, pairwise Welch test, we use a graphical tool of strip charts which helps us to identify a gene which has a distinct profile in one of these three groups. So what are these strip charts? It's a graphical representation of the data. So these are the strip charts. These are the strip charts corresponding to top 9 genes. By top 9, what I mean is the equality of means is rejected for these 9 genes, and for these 9 genes the p-value corresponding to Ebb test is smallest. Okay. So if we look at this first strip chart where the number 1 corresponds to the rank 1 gene, what we notice here is the 8 observations coming from NO group, that one stands for non-obese group, second is for obese group, and number 3 is corresponding to medium obese group. So here the first gene, rank 1 gene, has a different profile in all the 3 groups. If we proceed to rank 2 genes, the profile as far as sample shows is more or less same in the first and third group, but distinct in the second group. For the third gene, it is more or less same in second and third group, but slightly different at the first group. So we observe all these strip charts, and along with that, the p-values of the pairwise test. So here is a table of top rank genes. We know the gene identity, so we know the annotation or identity of a gene which has this distinct profile. These are the F values, so these are the top ranked genes according to one-way ANOVA F test. The p values are very small, so all these are significantly differentially expressed genes. The next column gives you the p value for testing mu1 equal to mu2. The next column is the p value corresponding to the pairwise test for mu1 equal to mu3, and next one is mu2 equal to mu3. For rank 1 gene, what we notice is the p value corresponding to F test is 0. So this gene has a distinct behavior in all the 3 groups. It doesn't have the same profile in the 3 groups. That is the conclusion from p value F test. Now if we look at the p values of these pairwise tests, these are 0.02, 0 and 0.002. So these p values and the strip chart tell us that, okay, this is a gene which has a distinct behavior in all the 3 groups. So mu1 is not same as mu2, mu1 is not same as mu3, mu2 is not same as mu2. It has a distinct profile in all the 3 groups. Look at the rank 2 gene. It's again, if p value corresponding to F test is 0.001, so it is again significantly differentially expressed gene. And if we again look at the p values of the corresponding pairwise t test, the p values are 0.001 for comparing mu1 equal to mu2. It tells us that mu1 is not same as mu2, but the p value corresponding to mu1 and mu3 is 1. So it tells us that these are Bonferroni adjusted p values. So it tells us that, okay, this gene, rank 2 gene, has more or less same behavior in first and third group. And also, that's also evident from our strip chart. 8 observations in first group and 8 observations in third group are more or less equally distributed. While the 8 observations in the second group are at a low level, so this is a gene which is expressed at high level in first and third group, but it is expressed at low level in the second group. So the p values and the corresponding strip chart help us to specify the profile of the gene in these 3 groups. So the profile we are written as plus minus plus. What it indicates that this rank 2 gene is overexpressed in non-obese group, it is underexpressed in obese group, and it is again overexpressed in medium obese group. Okay, so the appropriate test procedure, corresponding pairwise test procedures, and this graphical representation of strip charts help us together to identify the profile of the gene, how it behaves in these 3 types of genomes. So proceeding exactly on same line, if we look at the rank 9 gene, again this rank 9 gene has a p value 0.007 for one-way ANOVA F test. So it has a different profile in 3 conditions. If we look at the p values of the corresponding pairwise t test, the equality of means is rejected for group 1 and group 2, group 2 and group 3, but equality of means is accepted for group 1 and 3 because the p-value is 1. And if you look at the strip chart corresponding to this 9th rank gene, again the picture is more or less similar to 2nd rank gene. This has the gene expression values slightly at the higher level in 1st and 3rd group and at a low level in the 2nd group. So the profile of the gene is again plus, minus, plus. So this rank 2 gene and rank 9 gene are such that for these two genes the expression is at low level in obese group and at more or less similar level in the remaining two groups. So this is how one can decide the profiles of these top ranked genes, and then these are further investigated by biologists. So using exactly similar techniques, strip charts and the p-values of pairwise t-test, we can divide these top 45 genes, top according to one-way ANOVA test, in 6 groups. What are those 6 groups? The 6 groups which indicates the genes which have different profiles in the 3 groups. So rank 2, rank 9, we have seen already that they are the nature of the type plus, minus, plus. So 2, 9, 15, 17 and 40, these are the genes with these ranks which are expressed at a low level in obese group and equal level in the remaining 2 groups. The genes with ranks 13, 20, 24 and so on are such that they are expressed at higher level in non-obese group and at more or less same level in the next 2 groups. So this table is the most important summary of our analysis. This we can do for many more genes, and these genes are further investigated. One can verify these inference procedures, the results of inference procedures, by some other techniques also. There are a variety of multivariate analysis techniques. So I'll just tell you one technique. Okay, we'll come to this later. So this is a multivariate display of the top 45 genes. This is known as a biplot. The underlying mathematics for this is singular value decomposition, a very important tool from linear algebra. What it does is this entire data matrix of dimension 22,283 by 24 is reduced to a rank 2 matrix. It's a very important technique, and that rank 2 matrix basically can be interpreted as the collection of pairs which can be plotted, and you get such a plot. The importance is this plot shows the classification of your samples and classification of your variables as well. So what I did is I have selected the gene data matrix of top 45 genes and 24 columns. So my data matrix was of dimension 45 by 24. It was reduced to a rank 2 matrix and it was displayed. It's possible to display it this way. And now you notice all these O symbols are together at the at one part of this matrix. All NO symbols are together and all MO symbols are together. So we know that out of these 24 samples, 8 are corresponding to one group. All these 8 are together. NO samples, non-obese samples are together, and medium obese samples are together. Something more important is the genes with ranks 2, 9, 17, 14, 15. These are 2, 9, 15, 17 and 40. These are the ranks which are expressed at a low level in obese group. Okay. Now look at this obese group. The arrays which indicate to this obese group, if these are extended backwards, all these pass from these groups 2, 9, 17, 14 and 15. The interpretation is for all these genes with these ranks, the expression value is at a low level in plus group and they indicate towards this NO and MO group, so they have high expression values in NO and MO groups. Exactly on similar lines, look at these genes number 16, 21, 45, 22, which are expressed at high level in obese group. And if we again go to our table, expressed high level in obese group is this group number 4, minus plus minus, or this minus plus plus. So group number 3 and group number 4, where the genes are expressed at high level in obese group, these are grouped together and their arrows are corresponding to O group. 16, 21, 22, 45, 33, 11, 29 and so on. Okay? So this is a very important display of biplot. It tells us the grouping among observations, grouping among variables, and interrelation of observations with the variables. So, a very nice technique, mathematical technique of singular value decomposition helps us to display the group of genes and their relation with the various types of samples under study. Same thing can be revealed in dendrograms. So this is hierarchical clustering procedure. Use any linkage method, single linkage or complete average vaults. What you notice is that the 6 groups are again can be identified here. So this rank 2 genes, or let's look at this first group, rank 45, 23, 13, 8 and 12, these form one group. And if we go to our table, these 2, 9, 15, 17, 40 are in one group, and more or less they are there. No, sorry, I am wrong. Where is that? 2, 9, 2 is here. So the one can read this carefully, and again there are 6 groups are visible here also. One can also go for k-means clustering and identify this grouping of genes, grouping of observations. Our importance is grouping of genes because we already know the grouping of samples. So using this clustering techniques, this is essentially principal component analysis technique and inference technique, we are able to provide this information that genes with these ranks can be interpreted as marker for non-obesity. Genes with ranks 2, 9, 17 and 21 are sort of markers for obesity. We also have marker genes for non-obese group, and then these are possible markers or distinguish between these groups. This description is according to the magnitude of p-values and on the basis of strip charts. So, in summary, what we had done is out of 22,283 genes coming from 3 groups of samples, we are able to identify a handful of genes which are biomarkers for these 3 groups. Okay, so it's a exploratory analysis. Out of 22,283 genes, we identified 236 genes having a different profile in 3 types of obesity. We use strip charts and pairwise comparison to decide on the profile, whether it is overexpressed in one group or underexpressed in the other group. Of course, these results are, we use again using inference procedure. We always validate all these results using other tool. So we use multivariate analysis tools, and then the last stage, of course, this need to be validated by biologists using the RT-PCR techniques and so on. Here is a set of R commands again. So This MAP is a data matrix of quantile normalized columns. I will just explain few commands. That first command, scale T MAP. MAP is a data matrix. Rows corresponds to gene, columns corresponding to sample. That T stands for transfer, so we take a transpose of the matrix and we scale it. That scale means making each column a standardized column. In a sense, its mean is 0, variance is 1. So when we transfer it, the 24 observations are scaled in a manner that their mean is 0 and variance is 1. We again transfer it, so A1 is mean transfer of A. So we now have for our Shapiro test all the 24 observations in each of these rows to be on equal platform. And then we apply Shapiro dot test on this. A single loop carries out all these 22,283 tests in less than 1 or 2 minutes. So this R software is very powerful in that sense. And on the basis of ordering, we select the top genes. On the genes for which it is rejected, we apply Kruskal dot test. And again, on the basis of pairwise test and strip chart, we identify few genes and decide on their profiles. Similar loops are written for Bartlett test to test the equality of variances. Then one-way ANOVA dot test, when we say variance dot equal to false, it is one-way ANOVA usual, one-way ANOVA well-checked test, because equality of variances is not accepted. And another one is one-way ANOVA dot test, when variance dot equal to true, is the loop corresponding to one-way ANOVA. F test, when the equality of means and equality of variances and normality of the observations is also accepted. So this is a set of commands which helps us to analyze such a huge dimensional data set and arrive at the aim of identifying only a handful of genes out of thousands and thousands of genes. Okay, thank you.